Picking up where we left off, another organelle that's required for the job of, quote, making energy is the mitochondria, sometimes known as the mighty mitochondria. Its function is to make ATP by doing cellular respiration, actually transferring energy from food to the molecule of ATP. So the sugar plus the oxygen yielding ATP, which fuels the work of life. The structure of mitochondria, there's a double membrane. There's a membrane on the outside and a twisty membrane on the inside. And mitochondria, they look kind of like slippers in our diagram, but there's our mitochondria. Make ATP energy from sugar and oxygen. Plants actually make energy two ways. So plants also have mitochondria, just like animal cells do. They do cellular respiration, just like animals do. It's just that animals get their sugar by eating, whereas plants are going to get their sugar from doing a process called photosynthesis, and that's where chloroplasts come in. So they have a second structure called a chloroplast that make energy and sugar from sunlight. And they do this in the process called photosynthesis, where they take sunlight, water, carbon dioxide, and they turn that into sugar, so energy stored in sugars, as well as active energy in the form of ATP. So mitochondria are in both cell types. So we have an animal cell over here, and we have a plant cell over here. Animal cell has mitochondria, so do plants. Common misconception that I have seen students do is they think that mitochondria are animals only, but no, plants. Plants have mitochondria too, as well as their chloroplasts. So looking at a diagram of a plant cell, so if you flip your diagram over, you should have a plant cell on the backside. Plants have cytoplasm. They have a cell membrane, but they have a cell wall. This is something that animal cells do not have. They also have a central vacuole, something else animal cells do not have. They have mitochondria, just like an animal cell. They have chloroplast, which animal cells do not have. So the second job of cells is to make proteins. So the cell workers need these proteins to do everything. So making proteins to run life, growth, etc. We need to be able to read the genes, our DNA, build the proteins, so structural proteins, proteins that will function as enzymes, proteins that will be signals that will allow the cells to talk to each other. And the organelles that are going to do that are the nucleus, the ribosomes, endoplasmic reticulum, and Golgi body. So proteins do all the work of the cells. One of the major jobs of the cells is to make protein, because proteins do all the works of the cells. The instructions for building protein come from DNA. DNA is then copied into a molecule called RNA. That RNA message is going to leave the nucleus and be picked up by a ribosome that is going to actually assemble our protein. And then our protein actually is what makes most of the structural components of our cell, and the enzymes and the signaling molecules, as well as being receptors for communication. So the nucleus, the function of the nucleus, so this is a nucleus right here, is the control center of the cell, protects the DNA. The DNA has the instructions for building the proteins. The structure is that there's a nuclear membrane, also sometimes called the nuclear envelope, that encircles all of the DNA that's inside. So all these little squiggly purple lines represent DNA. Inside there is a structure called the nucleolus. That's this darker circle right here. And this is the ribosome factory. This is going to make all the ribosomes for the cell. The nucleolus makes the ribosomes. And the DNA is organized into sections called chromosomes. So if we go back to our diagram, here is our nucleus, which protects DNA and controls the functions of the cell. This is our nucleolus, which produces ribosomes. And here are the chromosomes that are the DNA instructions for building the proteins. So ribosomes. Ribosomes are protein factories. This is what actually assembles amino acids into a protein. And it does this according to instructions from DNA that gets copied into RNA. The structure, we have some that are free-floating in the cytoplasm. We call them free ribosomes. And we'll have some that are attached to the rough ER. So here, this kind of tan colored thing is the endoplasmic reticulum, and all these little dots that kind of look like beads, those are ribosomes. And if we zoom way in, this is an enlarged picture of a ribosome. You can see it has two halves, the larger subunit and the smaller. So if we look at our diagram here, all these teeny tiny little dots, you can see ones that are floating in the cytoplasm, those are free ribosomes, and ones that are attached to the endoplasmic reticulum, but these are ribosomes which build proteins. Endoplasmic reticulum, for short, we call it the ER. The function of that is to work on proteins. It actually folds and modifies proteins, helps complete the protein after the ribosome puts together the polypeptide. So this is going to help form our secondary and tertiary structure of our protein and even quaternary. It makes membranes. So there's actually two parts to our ER. There's a rough ER that has ribosomes and a smooth ER. The rough ER is what modifies the proteins. The smooth ER is actually for the synthesis of lipids. So the lipids, the phospholipids that'll make up membrane are actually produced by the smooth ER, and it's called smooth because it doesn't have ribosomes on it. The rough ER is called the rough ER because it's dotted with all of these little ribosomes, making it look or appear rough. Here's actually a transmission electron microscope picture. Here is rough ER, and you can see all the dots of the ribosomes, and then here's smooth ER over here. So rough ER has ribosomes attached to it, works on the proteins. Smooth ER makes membranes. So here's where they are in our diagram. So all of this structure in here, highlighted now in red, is our ER. So the rough part works on proteins, and the smooth, which is over here, is going to make membranes, making lipids for the membrane. Golgi. So Golgi body finishes, sorts, labels, and ships the proteins. This is what's going to tell our protein to go to its final destination. Think of it kind of like the Postal Service or the UPS headquarters of the cell. It's going to package our proteins up and tell the proteins where they're going to go. It ships the proteins in little vesicles, so each of these little circles are vesicles made of membrane that have pinched off the Golgi. So the structure, a bunch of folded membrane sacs filled with enzymes. So right here, this little guy here in yellow that's now highlighted in red, that is our Golgi, which finishes, packages, and ships our proteins to be exported from the cell. So looking at this whole process of making proteins, right here is our nucleus. Within the nucleus, we have DNA, and DNA is copied into a molecule called RNA. That RNA is going to leave the nucleus. The process of copying DNA into RNA is called transcription. We'll do a whole unit on this later. The RNA is then going to exit from the nucleus through a nuclear pore, and it's going to be picked up by a ribosome. So all of these little stacked guys are ribosomes, and these ribosomes are sitting on the larger structure, the endoplasmic reticulum. The ribosome is going to pick up the RNA, so a ribosome with its RNA. The ribosome is going to read the instructions of the RNA to assemble amino acids together into a protein. So here, each of these little squares represent an amino acid. This is our polypeptide, which is our primary structure for our protein. This process is known as translation. Transcription and translation, we'll talk both about these later during our genetics unit. Then this polypeptide, the primary structure, is going to move its way through the ER, and as it does that, the ER is going to work on it, fold it, modify the protein, and then it's going to put it into a vesicle. So right here is a vesicle. This vesicle is going to transport our folded protein to another region of the cell called the Golgi. So the vesicle is going to travel to our Golgi body. And so the Golgi body is going to do even further modifications to our polypeptide, or folded polypeptide, and create our final form of our protein. And as it's modified, it's going to be finally packaged into a vesicle. So the Golgi is going to put our final protein into a vesicle. And that finished protein is going to travel via vesicle out of the cell and into up here the bloodstream. So the final destination of this protein is to travel through the blood to somewhere else in the body. The protein is on its way. Hooray! So we have a lot of these structures also in a plant cell as well. So there's the nucleus in a plant cell. There's the nucleolus to make the ribosomes. There are ribosomes. They're kind of hard to see in this diagram, but the little teeny tiny dots, those are the ribosomes that are sitting on the rough ER. There's endoplasmic reticulum in a plant cell. There's rough ER and there's smooth ER. There's Golgi within a plant cell as well. So the last thing that cells need to be able to do is make more cells. So making more cells, we have to do this to replace cells that die or cells that get damaged or to allow you to grow. And in order to do this, you have to copy the DNA, make extra organelles so that each new cell has all the equipment it needs to survive. And then you have to divide the DNA and the new organelles between the two new cells, which we call daughter cells. So here is our cell. These little X's represent chromosomes or the DNA. And then these little purple lines are going to represent something called the cytoskeleton that will help to divide our chromosomes. I'm going into each new cell, so one daughter cell, two daughter cells, and each of them have a full copy of the chromosomes. So the organelles that do this work are the nucleus, centrioles, and something called cytoskeleton. By the way, the prefix cyto, anytime you see that, cyto means cell. So this word actually means the skeleton of the cell. So centrioles, their function is to help coordinate cell division. Find these in animal cells only. There's one pair in each cell. So the centrioles would be these black dots over here. And the cytoskeleton that they're organizing are these green lines, and the chromosomes are these little X's in the middle. And what happens is these cytoskeleton strands will shorten and pull apart the chromosomes, just like that. Watch, I'll do it again. So it's going to pull the chromosomes apart. One copy of the DNA goes to this new daughter cell, and one copy of the DNA goes to this daughter cell. Cytoskeleton, the function of this is to provide structure and support for the cell, help separate the chromosomes, move vesicles and organelles within the cell, and it's made up of a network of protein filaments and fibers. There are actually three types. There are microtubules, microfilaments, and intermediate filaments. And again, they help coordinate the movement of materials within the cell as well as maintain the structure of the cell. There you go. Cytoskeleton pulling apart the chromosomes. So on this picture, you can't really see the cytoskeleton. That's a sad thing on my part, but there's so much of it. If we put it in here, the cytoskeleton would actually obscure your view of all the organelles. It actually fills pretty much the whole inside the cytoskeleton. But what you can see, there are centrioles. These do not exist in a plant cell, so don't look for it on your plant diagram, but they help coordinate cell division. So cell summary, cells have three main jobs: make energy, so need food and oxygen to do this, does cellular respiration and photosynthesis if it's a plant, need to remove the waste, for making proteins, need instructions from DNA, need to chain together amino acids, finish them up, and then ship them out, and then for making more cells, need to copy the DNA and then divide it amongst the daughter cells. So that concludes part 2. Please continue and watch video number 3, which will talk about cell junctions and talk about cytoskeleton in more detail.